molecules of pyruvate. And we're going to proceed to a series of reactions which actually run in a cycle. You'll see why it is a cycle in a second. It's commonly referred to as the Krebs cycle, named after the person who elucidated it, but is more properly called the citric acid cycle, because citric acid is the very first product of the whole cycle. And so that's what we're going to see. We're going to see how this whole system here works in the second stage. This is very, very productive, not in terms of ATP. It does produce a little bit of ATP, but not much. But it is extremely productive in terms of electron carriers. Look at this. It produces a total of 10 molecules of NADH in all of these reactions, and 2 molecules of another electron carrier called FADH, which is very, very similar to NADH. So this produces 2 molecules of FADH2. And all of these are going to be taken to another set of reactions, which we're going to discuss in some detail, called oxidative phosphorylation, another kind of phosphorylation. So, and this oxidative phosphorylation is fantastically efficient in producing ATP. It's going to produce 34 molecules of ATP just from this process here. 36 molecules altogether, counting these here. And 42 actually, if we counted everything from glycolysis, which doesn't actually happen. Okay, so we're now going to watch as we proceed through these reactions. We're going to follow pyruvate. Please remember, there are actually 2 molecules of pyruvate produced from glycolysis, from the process of glycolysis. Glucose is 6 carbons, pyruvate is 3 carbons. So it's 2 molecules comprised of 3 carbons. This whole process is also referred to as cellular or aerobic respiration. And that is because in order to proceed, it requires the presence of oxygen. And the first steps actually that we're going to see, the oxygen is not performing exactly the same role as it does in the last step, but nonetheless, the whole system is driven by the presence of oxygen. And the reason for that is oxygen is an extremely electron-negative element. It pulls electrons towards it. And this is one of the driving forces for the whole system. So the first stage, the first set of reactions for aerobic respiration that we're going to talk about are largely concerned with this entire cycle here. I actually got a bigger, this is exactly the same. It's just a larger version. So here's our pyruvic acid 3-carbon compound here. We're going to push this through what is called a preparatory step, or an intermediate step here. The pyruvic acid here is combined with a compound called coenzyme A. Now, think back and you'll remember that the coenzymes are substances often derived from vitamins, which are associated with enzymes. And here is one specific one called coenzyme A, features very prominently in all of these reactions. Coenzyme A is enzymatically combined with NAD, with the pyruvic acid, sorry. And one of the sets of hydrogens is stripped from the pyruvic acid and is added to NAD+ to form this electron carrier here, NADH. So we end up with this 2-carbon compound here, combined with the coenzyme A. And this is called acetyl coenzyme A, and that's a name you must remember. acetyl coenzyme A, 2 carbons, and the coenzyme A. What happened to the carbon, extra carbon? There was 3 carbons here. Breathe out. What are you breathing out? You're breathing out carbon dioxide. One of those carbons comes from this whole reaction here. Stripped off the pyruvic acid, is combined with oxygen, and you breathe it out as carbon dioxide. So this is acetyl coenzyme A. Acetyl coenzyme A is important because it's the only way that we can get this compound into this cycle here. It enters the cycle by combining with this substance here called oxaloacetic acid, and it forms citric acid. The coenzyme A is stripped off, and can be reused, can be recycled. And instead, with oxaloacetic acid plus the acetyl part of this forms this compound here, again a 6 carbon called citric acid. 1, 2, 3, 4, 5, 6. This is why this is often called the citric acid cycle, also called the Krebs cycle, but more properly it's called the citric acid cycle because citric acid is its very first product. Remember, each step is enzymatically governed. We're not going to go into detail about every single step in this cycle. You'll be glad to know. But I do want you to remember what is actually produced. First of all, carbon dioxide from the intermediate step here. And 1 NADH there. NADH and carbon dioxide. In the cycle itself, here's NADH produced here and another carbon dioxide. We've stripped off yet another of the carbons. Each time this happens, the hydrogens are being stripped off and harvested and put into electron carriers. Here's another step where the same thing happens. We're going to strip off hydrogens, add them to NAD+, form NADH. This is another step at which carbon dioxide is produced. Realize that what is happening is we put this pyruvic acid, this 3 carbons, we added it. We first of all stripped off, ended up with 2 carbon. We added this to this oxaloacetic acid. So each time we lose a carbon from that, we're losing one of our original carbons from here. We're going to end up with a 4 carbon compound, which we're going to feed back into the cycle. Okay. At this stage here, when we move from here to from this compound to this compound, we're actually going to form a small amount of ATP. Again, substrate level phosphorylation. So this ATP is formed here. This stage here, we form another electron carrier, FADH2. We proceed with, at this stage here, we strip off more hydrogens and form more NADH. And what we end up with through this whole cycle, we end up back with a 4 carbon compound, oxaloacetic acid. That's why this is a cycle. We started with oxaloacetic acid and we regenerate it in the cycle. In the process, we carry along these 2 carbons, which we pump in from the pyruvic acid. We pump those in, strip them off, and at the same time strip off all the hydrogens that we can, and we form these electron carriers, NADH, NADH, FADH2, NADH. And remember something, this cycle runs twice for every molecule of glucose, because from every molecule of glucose, we produce 2 pyruvic acids, which enter the cycle. So we end up now, we've completely broken the glucose. There are no carbons from the glucose left. You have breathed them out as carbon dioxide. Okay, they're gone. But what does remain are all of the hydrogens. All of the hydrogens that were associated with that glucose molecule are now bound to electron carriers there, 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 and there. And some of their energy has also been bound into ATP. But it's these electron carriers now that we are really, really interested in, because we're now going to follow what happens to those electron carriers, and we're going to find out that the cell now can use these in an extremely efficient system to produce a lot of ATP, not these tiny amounts that we produce in glycolysis, and here just a little tiny bit in the Krebs acid cycle. The point of the cycle is not to produce ATP, though it does produce a little bit. It's to produce these electron carriers. That's what is going to be really critically important. The next stage of that we're going to watch are the processes of aerobic respiration or cellular respiration. And these, all of these processes depends entirely upon the presence of sets of substances which are embedded in the membrane. The other processes, the citric acid cycle and glycolysis, can take place inside the cytoplasm or inside the interior of an organelle. But the processes of aerobic respiration, of cellular respiration, are absolutely dependent on the presence of a membrane, for reasons that will become apparent. The first one being that is entirely dependent upon proteins and other substances which are embedded in the membrane. They are held tightly in the membrane. The second reason the membrane is critical is because it is essential that conditions on one side be different to conditions on the other side. Remember that I told you before, that is one of the most important functions of membranes in general. The conditions can be kept different on one side to what they are on the other. The other thing to understand about this system is that when we look at all of the substances which are embedded in the membrane, we need to realize that they are all arranged in a redox chain, a chain of substances which will sequentially perform oxidation reduction reactions. One substance, we're going to pump in electrons at the very beginning. One substance is going to be reduced. It is then going to be oxidized. And how do we reduce a substance? We add electrons. How do we oxidize a substance? We take away electrons and donate them to something else. That is what's going to happen. We're going to pump in electrons here. They will temporarily reduce the substance, which will then be oxidized by the next substance. So they will give up those electrons and donate them to the next substance. The same thing happens. It is temporarily reduced, then it is oxidized by the next substance, which picks up the electrons. And it picks up hands them on, hands them on, hands them on, hands them on, hands them down a whole chain through a whole series of these redox reactions. And all the way along the line, every time this happens, the electrons give up some of their energy. Some of their energy, at least, can be used to work. And we'll see now what that work actually is. But all the way along the line, some of the energy, not all of it, because no reaction is 100% efficient, all the way along the line, the work is being done as these electrons cascade through all of these substances. At the very end of all of this, first of all, we can't just have electrons flying around the cell willy-nilly without electrons don't do that. Okay, they only temporarily can be separated from their protons. Ultimately, the electrons have to be, something has to happen at the end. At the end of all of this, we have to have a final substance which is going to accept the electrons as as a final destination. And that we call the terminal electron acceptor. And the terminal electron acceptor in this whole system is oxygen. Oxygen is extremely electron negative. And in fact, once the electrons enter this whole system, they are pulled through by the attraction of oxygen. And ultimately, they're going to be bound to the oxygen. There's another player here that we haven't talked about. If I take a hydrogen atom and I strip off its electron, even temporarily, what am I left with? I'm left with H plus, right? H plus, a proton. So we have to think about those protons as well. If we're stripping off electrons, we are creating protons. And those protons are also extremely useful to the cell. This whole series, these here in these substances embedded into the membrane. Here's another view of it, which we'll see again. I'll put up a bigger version. These substances embedded in the membrane are carrier molecules. So they carry the electrons temporarily, cascade them through. But the energy which they harvest is used. And it is used because, and the way it is used is that several of these substances are actually pumps. They are transport proteins. And what they transport are protons. They transport protons. They pump protons. Every time electrons pass through them, they use the energy of those electrons to pump protons across one side. This means that on one side of the membrane, if we're adding protons, what are we doing to the pH? We're dropping the pH. The pH is much lower on one side of the membrane than it is on the other side. The other thing that has happened is we've actually taken a chemical, a proton, and we're accumulating it. So we have a concentration gradient of protons. Not only that, we also have a charge differential. We have protons, more positive charges on one side than we have on the other side. The one side is much more positive than it is on the other side. There are far more electrons on this side than there are on that side for a start. So we've got positive charges on one side, negative on the other. We've got all sorts of forces wanting to be equalized. That, across the membrane, that is potential energy. A very powerful potential source of energy to the cell. And we'll see exactly how the cell uses it. This is a chemiosmotic difference. It's a difference in concentrations, but also in charge and also in the nature of the substances which are on either side. We refer to it as a chemiosmotic differential. Okay, so let's have a look at it in a larger diagram and in a little bit more detail. Again, I don't expect you to remember all of the names and everything. I'm more interested that you understand the principles of what is involved. Just let me check one thing, please. Okay, fine. All right, so the first thing, let's orient ourselves. Here's the membrane. Here it is here. In prokaryotes, this is actually the outside. This is the outside of the cell. This inside in here, this is the cytoplasm, okay? Here's the membrane, and embedded in the membrane are all of these substances, different substances. There are more of them actually than are shown here, but they form a carrier system. The electrons are brought to this carrier system by the electron carriers, for example, by NADH, but also by this other substance called FADH2, which this is nicotinamide adenine dinucleotide. This is flavine adenine dinucleotide. They're very, very similar in substance, and both of them are electron carriers. Each of them is carrying 2 electrons. Okay, so we're bringing in a pair of electrons. This first substance here is a very, very powerfully attractive substance for electrons. And it very easily oxidizes the electron carrier. So we're going to oxidize the NADH. We're going to strip the electrons and protons off it. And this carrier is going to bind the electrons temporarily. It's going to be reduced. So this carrier is reduced. Those electrons are then going to be handed off, and they're going to be handed through this whole chain of substances all the way down. There are 3 that we are particularly interested in, and it is this one here, this one here, sorry, and this one here. This one, and this one. Because these are complex, very complex protein substances which function as proton pumps. So they can actually take protons and pump them across. Where are those protons derived from? They derive from the fact that we're stripping electrons off hydrogens. We're stripping electrons off hydrogen, sending the electrons through, but we're not sending the protons through here. Instead, the protons are being accumulated on this side. And these proton pumps actively pump them across, pump them there, and pump them there. So the cell begins to accumulate protons on this side. That means the cell is accumulating both a chemical, the proton itself, but it's accumulating charge as well, and it's dropping the pH on this side relative to the pH on that side. This creates a drive to equalize all of these forces across the membrane, but the membrane is a barrier. It's a barrier. I've said this to you before. If I've got a barrier, an insulator, right, and I accumulate charge on one side, which is different to the charge on the other side, I've got positive charge on one side, negative charge on the other side, and a barrier in between, what have I created? What I've created is a battery, just like the battery that you use in a flashlight or anything else. That is exactly what a battery is. It's a substance or a set of substances which allow negative charge to be accumulated on one side relative to a positive charge on the other side. That is potential energy. They cannot equalize much as they want to because there's a barrier in between them. But if I connect them somehow, the electrons will flow from where they're accumulated to the positive. In other words, the charges will equalize. And that flow of charge of any sort can be harvested to do work. And that is exactly what the cell is going to do. The cell is going to accumulate these positive charges on this side relative to this side here. At the same time, there's also chemical differential and everything else. Huge potential energy across this thin barrier of the membrane. And if we find a way to equalize those, we could harvest the energy as they equalized and use it to do work. So here's exactly what happens. We have all these substances in this carrier system embedded in the membrane. We introduce electrons. We actually can introduce it at 2 different places. The NADH introduces here, the FADH2 introduces them here. The electrons flow through this whole system. As they do, they cascade down this series of redox reactions, and the electrons actually lose energy, and that energy is harvested, and it's harvested at 3 places, there, and there, to pump protons across. The electrons stripped off the hydrogens means that we start to produce protons here. The cell pumps those protons across and accumulates them outside here. Now at the end of it, the electrons now can't just go free. They can't just go across the cell. they have to be bound to a terminal electron acceptor. And what they are bound to are protons. So they're bound to protons and to oxygen. Here's the oxygen here. Oxygen is actually the driver. It's pulling all of these electrons through. And the electrons are finally bound to the oxygen and to protons to produce water. And there it is there, breathe out. What are you breathing out? You're breathing out carbon dioxide. Where did that come from? It came from the intermediate step and the Krebs cycle, the citric acid cycle, right? That was where all the carbons were stripped off. Now, all of the electrons were used to do work to produce ATP. We'll see exactly how in a minute. But the electrons in the end are bound to oxygen and to their protons. They finally meet up with their protons again to form water. And that is a byproduct. That is the other byproduct, carbon dioxide and water. You breathe them out as a result of these processes of aerobic respiration. So that's fine. Now our electrons have come screaming all the way through. They've given up energy which the cell has harvested, and it's harvested it by creating this huge potential energy across the membrane. How is it going to use that energy? Well, it does it with a spectacular set of enzymes which are all combined together to form this major enzyme here called ATP synthase. As soon as you see that, you see the ASE, you know it's an enzyme. And a synthase is something which creates, it is something which creates the substance. So this is ATP synthase, and it is making ATP. How does it do it? It is a channel protein. It allows protons to flow through it. Remember, these protons accumulated here on this side. What they wanted, what the drive is for them to end up back on this side. Why? Because there's a charge difference. They've accumulated positive charge out here. It's more negative in here. They're going to want to equalize. Second of all, it's a chemical difference. These are actual chemicals, these protons. It's actually hydrogen. It's a hydrogen ion, H+. It's a hydrogen ion. This has been accumulated here, and it's lacking here. It's going to equalize on that side. So there are powerful chemiosmotic forces driving protons. As they move through this ATP synthase, they lose energy. And that energy is harvested to produce ATP, and to produce it in quite high quantities. In actual fact, they produce about 32 molecules of ATP for every glucose molecule. For all of the electrons from one glucose molecule that pass through here, we create about 32 molecules of ATP. So that's how this whole system works. All of the original things, glycolysis, the intermediate step, the citric acid cycle, all of those things that created a small amount of ATP, but what they really, really did was they loaded up electron carriers, NADH and FADH. They loaded them up with electrons so that those electron carriers could donate electrons to the system of electron carriers here embedded in the membrane that now create these differentials across the membrane and then use that potential energy to actually create ATP here. So this is called aerobic respiration for the simple reason that the terminal electron acceptor, the final electron acceptor, is oxygen, is molecular oxygen. And that is the reason that we are so dependent on oxygen. Obligately aerobic organisms organisms which require the presence of oxygen in order for their metabolism to function properly are called obligate aerobes. There are many organisms which can function anaerobically. And they can continue to function in the absence of molecular oxygen because they can come to rely, they shut down, they either don't have or they shut down these pathways that depend on oxygen, and they just rely on processes like glycolysis to produce a small amount of ATP to keep their systems going. So this is just a more elaborate version of the same thing. The reason I do want to point this out, though, just realize that this has been flipped relative to this diagram here. Okay, don't get confused. This is the outside. Okay. This is the cytoplasm here in this one. Again, just a series of electron carriers here, but 3 of those electron carriers are also proton transporters. And here, as the electrons flow through, this one here, this one here, and this one here, all transport protons. Just a couple of things of interest. The substance cyanide, I mentioned before when we talked about competitive inhibition of enzymes. Cyanide is a very dangerous substance because it is a competitive inhibitor for this enzyme here, for the last of these substances, which are called the cytochromes in this whole chain of substances, of electron carriers. Cyanide actually blocks the activity of this enzyme here. If it blocks the activity of that enzyme there, the entire pathway shuts down. And so aerobic respiration completely fails in the presence of enough cyanide to block all of this. That's why cyanide is a poison. That's why it is so dangerous. Other things to remind you about, first, and that is all of these electrons streaming through these substances, they have to have a final destination. Ultimately, they've got to be bound back to their protons. And they are bound back by forming water. We take oxygen, we take the electrons, and we take the protons which are flowing through the ATP synthase, and together they form water. This substance, ATP synthase, is extremely complex. It actually, as the electrons move through it, it's induced to spin. It spins around, and every time it cranks once around, it creates 3 molecules of ATP. Round, 3 ATP, round, 3 ATP, etc., etc. Yeah, you absolutely don't need to remember all of the details of its structure and everything. Just remember that it functions because it allows the passage of protons, and in doing so, it spins around and it creates ATP from ADP. It actually phosphorylates ADP using the energy from the electrons passing through. All right, so that is just a summary of the whole thing. So there are just a couple of sort of kind of complications, I guess one would say, in the whole story, and you'll be used to the fact that these very complex systems often do have complications associated with them. In order to explain this, I just need to come back here and re-emphasize the importance of this molecule here, of the oxygen molecule sitting here. Oxygen is important, first of all, because it's this very powerful electron attractor. It attracts electrons through the whole system. But second of all, it's the terminal electron acceptor. And together with the electrons, then together with the protons, it forms water, which we breathe out as a waste product. So free oxygen, molecular free oxygen, is essential for the functioning of that entire pathway. If it's not present, then the pathway cannot work. However, there are some organisms which are able to continue operating even in the absence of free molecular oxygen, O2, provided there are ions present which have oxygen bound to them. This is not all prokaryotes. There are only some of them are able to do this. But they can, for example, use nitrate, NO3, or nitrite, NO2, as a source of oxygen. They are going to strip off the oxygens here from these and use them as terminal electron acceptors. This is much less efficient than just if they were able to do it aerobically, but it's extremely useful because prokaryotes are very frequently put into anaerobic conditions, especially during, for example, during rapid growth. Prokaryotes use up the oxygen available to them very, very quickly, and conditions can go anaerobic very, very fast. And an organism which is able to do this, these are common ions, and an organism which is able to use oxygens bound like this is at a considerable advantage. And there are several of them that do it, and you will encounter some, I'm sure, in your lab exercises, because the testing for the ability to do this, to actually reduce nitrate or nitrite, this is a very common discriminatory test in clinical microbiology. It's something we do commonly in the lab when we want to classify organisms. So this is referred to, has a strange name, is anaerobic respiration. And it sounds like a tautology, but it's not. It's still respiration because they are using oxygen, but they're using it under anaerobic conditions because they can strip the oxygen off these oxidized ions. Okay. Right, so let me just go back here again, because there's another thing that we need to understand. And I'm going to go all the way back here to glycolysis, to remind you about something. And that is that I told you that some organisms are able to shunt to glycolysis in the absence of oxygen. If oxygen is present, they can proceed through cellular respiration, aerobic respiration, quite happily. If we take away the oxygen, then they are able just to rely on glycolysis. In addition, there are organisms which absolutely cannot use oxygen. They are totally intolerant of even of the presence of oxygen. And so those we refer to as being anaerobic systems. Some organisms are able to shift between the one and the other. Those we call the facultative anaerobes. Others require that no oxygen be present for them to metabolize at all. Those are the obligate anaerobes. They all depend on glycolysis as their source of ATP. So they go through glycolysis, and for every molecule of glucose, they harvest a net gain of 2 molecules of ATP. Not very much, but it is enough for their metabolism to proceed. The metabolism usually proceeds much, much more slowly than it would under aerobic conditions. But nonetheless, it's good enough. And there are many organisms which depend on this quite happily. But there is a major problem, and it lies not in the production of ATP, but this here. These electron carrier systems are involved in many, many different metabolic reactions, because this is a way in which we activate many enzymes, is by having them interact. That is why these are coenzyme systems. These electron carriers are very often coenzymes. So the cell actually needs a continual supply of NAD+, in the cell in order for systems to continue to work properly. The process of glycolysis, by its very nature, continually produces NADH and uses up all of the cell's stores of NAD+. And those reactions which require the presence of NAD+, and there are many of them, now start to fail because they don't have enough NAD+ to continue. Everything gets bound up into NADH, into the reduced form. This is a huge problem for the cells. And there are many strategies to get around this. And almost all of them depend upon taking this substance at the end here, the other product of glycolysis, which is pyruvic acid, and doing something with it, actually reducing it to produce more NAD+. If I reduce NAD+, I produce NADH. If I take this NADH and I oxidize it, to reduce another substance, I will regenerate my NAD+. And that is what many cells have different strategies for taking the products of glycolysis and reducing them to produce NAD+. So let me flip forward to here and show you this is exactly what the cell needs to do. Glycolysis here is producing ATP, that's fine, but it's also using up all the NAD+ and to producing NADH. The cell now needs to take that NADH and it needs to oxidize it. It needs to oxidize it to NAD+, to feed that NAD+ to reactions which require it. And the cell does this by taking pyruvic acid or something derived from pyruvic acid and actually reducing it. If it reduces it, takes the hydrogens from there, electrons in other words, from there, adds them to this substance here and produces some other substance. This is the process of fermentation. You will see sometimes glycolysis incorrectly referred to as fermentation. Glycolysis itself is not fermentation. Glycolysis is glycolysis. It produces pyruvic acid. Fermentation is a separate set of reactions which take the products of glycolysis and reduce them in order to regenerate NAD+. That is the purpose of fermentation. So the way there are many strategies which different prokaryotes will use in order to do this. But in general, we can kind of divide them into two systems. The first are a system of acid fermentations, where it's largely, most commonly and predominantly lactic acid that is produced, sometimes only, and those we refer to as homolactic fermentations. But very, very often in other organisms, they produce a mixture of different acids. Sometimes they're actually the other acids are actually more predominant than lactic acid. And those we refer to as the heterolactic fermentations. They produce lactic acid, but also other compounds as well. The other types of fermentation are alcoholic fermentations. And the most familiar alcoholic fermentation is that performed by yeast, which is a reflection of the fact that these systems can also be used by some eukaryotes. Many eukaryotes in their cells, including us, have fermentation pathways for those occasions when oxygen becomes limiting. In us, in human beings, and mammals rather, the clearest indication of this is in our muscles, where if we overwork our muscles and they become deprived of oxygen, they can continue to work for a certain length of time, only for a certain length of time, by resorting to metabolic reactions which are very similar to glycolysis and which actually produce lactic acid. It's the accumulation of lactic acid in your muscles that causes the muscles to burn when you overwork them. And it's one of the symptoms of tiring of the muscle. The most familiar alcohol fermentation is in yeasts, particularly in one strain of yeast called brewer's yeast. And those are the fermentations which we actually use in cooking and in brewing. And the reason for that is that these alcoholic fermentations produce two things. They produce ethanol and they produce abundant carbon dioxide. And in baking, for example, we make use particularly of the fact that yeasts produce lots of carbon dioxide. That is what causes bread dough to rise. The carbon dioxide accumulates and it inflates the dough. Ethanol is produced from yeast fermentation, and that is what we use in brewing. And there's a complex set of reactions to do this, which we are not going to discuss in detail. But just remember, they're predominantly, we'll see some of the other reactions in a second. Predominantly, there are these acid fermentations, the lactic acid fermentations, one sort or another, and the alcoholic fermentations. All of them take pyruvic acid. They're actually reducing pyruvic acid or its products in order to create NAD+. In the process, they produce a wide range of different kinds of byproducts of this process. So, for example, lactic acid is very common, very, very commonly produced. And the other is typical Saccharomyces. Saccharomyces is yeast, that is ethanol and carbon dioxide. There are some strange products. Some organisms, especially obligate anaerobes, can produce a very strange substances. For example, propionic acid, acetic acid, even hydrogen, all of these kinds of things are produced as byproducts. Clostridia, the Clostridia, which we'll hear about when we talk about bacteria and health, the Clostridia also produce many strange products. Butyric acid, very, very useful alcohols and industrial solvents. These are like butanol, acetone, isopropyl alcohol, all of these sorts of things. These are really, in all of these here, are very, very interesting because they can be harvested industrially. We can actually culture bacteria specifically so that they produce these products for us and then extract the products and use them. This is done on a wide scale to extract many of these different compounds. Some of the common bacteria that you will see in lab are facultative anaerobes, and when you grow them in the lab, they will produce Escherichia coli, which you will commonly deal with, produces produces largely lactic acid. Others produce ethanol as well, succinic acid. Some of them produce a mixture of these different acids. Some of them only produce one or two. Then there are a whole set of bacteria which do not produce acids at all, only produce different kinds of alcohols or other neutral substances. And this can also be an important distinguishing feature, clinically distinguishing feature in the lab between one kind of bacteria and another, that we have to distinguish what kinds of products they produce in the course of fermentation. Okay, so we have discussed two major ways in which the cell produces ATP, the most biologically useful form of energy in the cell. The first one was substrate level phosphorylation. And that is the enzymatic addition of phosphate to ADP to create ATP. And that is in the absence of oxygen. Okay, those processes proceed, for instance, in glycolysis, etc. The other system we looked at was aerobic respiration. That is that whole membrane-bound system driven by the presence of oxygen, finally acting as a terminal electron acceptor, which creates water, and in the process creates ATP. So those systems, the system of aerobic respiration is extremely efficient in producing ATP. Oxidative phosphorylation, finally, when we add up all of the ATPs, it is around about 32 molecules of ATP are produced for every molecule of glucose. So by far the most efficient of all of it is the system of membrane-bound system of aerobic respiration. And there is one last way in which organisms are able to produce ATP, and it's probably in many ways one of the most important of all, because it is the system on which all life on Earth depends, ultimately. And that is the system of photosynthesis. And photosynthesis is the manufacture of ATP by photosynthesis is referred to as photophosphorylation. That is phosphorylation of ADP by light. But the formation of ATP in photophosphorylation is actually one of the lesser aspects, less important aspects of the process of photosynthesis, as you'll see. The aim of photosynthesis, if you want to think of it that way, is not to produce ATP, although it does produce some ATP. The aim of photosynthesis ultimately is to produce biological molecules which contain energy, which has been produced from the conversion of light energy into chemical bonds, into the energy, potential energy locked into chemical bonds. I am only going to discuss photosynthesis extremely briefly, very, very, very briefly, because I have set a series of exercises for you to do, online exercises on photosynthesis, which comprise a major part of your next class, okay, which will be on Wednesday. So I'm only going to mention briefly here the major points about photosynthesis. The first thing is that photosynthesis also depends upon membrane-bound systems for the same reason. First of all, the enzymes must be bound into the membrane itself. But second of all, they utilize the fact that the membrane is going to form an actual barrier between inside and outside. And so the way in which this system is going to work is in some ways similar to that of aerobic respiration, because what we're going to do is going to use the energy of electrons. We're going to use the energy of electrons to drive the pumping of protons. But something other than that is going to happen during this whole process as well. So the next thing to realize about photosynthesis this is different to the process of aerobic respiration is that the terminal electron acceptor in photosynthesis is not oxygen, as it was in aerobic respiration. Instead, it is this substance here, NADP. Now that should ring a bell, because NADP is another of the electron carriers. And that's what we're going to be really interested in here in photosynthesis. What is really, really significant in photosynthesis is the creation of this NADPH, reduced NADP. And that is what really the whole point of photosynthesis you're going to see is going. Along the way, photosynthesis does produce some ATP. That is true, and produces it by the process of photophosphorylation. So let's have a look just at the very basics of photosynthesis. The first thing to understand is that photosynthesis depends upon the presence of pigments in the membrane. There are two pigment systems in the membrane, embedded in the membrane, here and here. And these, the most important pigment here is the pigment chlorophyll. There are different species of chlorophyll, by the way, and there are two species used in this form of photosynthesis. So the first thing is we have pigments. What is a pigment? It's a substance which interacts with light. Okay? It absorbs certain wavelengths of light. It absorbs the light, and in doing so, it has some associated with, the pigment has associated with it some electrons which are very loosely bound to the pigment molecule. And those electrons can be excited by the energy from light. And they can be boosted. They are circulating around molecules in orbit, at a very loose orbit. And when excited, they can be boosted out of that orbit and can actually be induced to reduce substances nearby. In other words, we can transfer, if we excite those electrons enough, we can get them to reduce substances close by to them. So that is exactly what happens. Here's a pigment molecule. Here, it's a chlorophyll. Light shines on it, and a pair of its electrons get extremely excited, and they actually reduce substances nearby. Then now the electrons start to cascade. They cascade now through a similar system where they oxidize and reduce one another to what we saw in aerobic respiration. The electrons cascade down, and just as before, first of all, we're creating protons. That's the one thing. But we have a proton pump, which harvests some of the energy to pump protons. And it's pumping protons to this side here. So there they go there. Where are those protons derived from? Well, this is one of the most fascinating things about photosynthesis in itself. And this is something which we still do not entirely understand. These protons actually derive from water. What happens in this system here, when this electron here is actually derived from a water molecule, they are stripping electrons off the hydrogens associated with water. We haven't got electron carriers coming in here the way we did in aerobic respiration. We have to have a source of electrons, and the source of electrons in photosynthesis is actually the water molecule. Electrons are stripped off water. We say that water here is cracked. We strip off electrons, we leave protons, and we create free oxygen. That is why oxygen is a product of photosynthesis. It's a waste product of photosynthesis. Breathe in, and the oxygen, most of the oxygen that you are breathing in is actually the byproduct of photosynthesis. It's byproduct of photosynthesis, just this molecule here, this reaction here, doing all of these incredibly significant things. It is cracking water. It's stripping hydrogen electrons off the hydrogens. It's making protons on this side here. They're also being accumulated on this side, by the way, because they get pumped across. And these electrons are being cascaded through this electron carrier system. And in doing so, they're being induced to do work. And that is to pump more protons across. So we're going to accumulate protons on this side of of the membrane. This pair of electrons here, it actually gradually lose energy. And when they, at one point when they have lost a lot of the energy, they're actually donated to another pigment molecule here. This is the second chlorophyll molecule. And the second chlorophyll molecule can also be excited by light. And this pair of electrons now is boosted again and is shunted off through another set of electron carriers and is donated to NADP. And together with protons, some protons from this side, it's used to form NADPH. This is an electron carrier. It has a huge amount of energy associated with it. And that energy has been acquired from here. It is energy which has been acquired from light shining on these pigments. And it is now being converted into the energy of a chemical bond. And it is lodged in this electron carrier, NADPH. And we're going to see how the cell now utilizes that. So that's the first set of reactions that is taking place, okay, is to create this set of electron carriers. And it's all depending on light shining on pigment molecules and exciting their electrons. In addition, we accumulate protons on the side of the membrane. And we have in this system exactly the same set of enzymes, ATP synthase, which allows the protons to flow through the ATP here and create, allows protons to flow through this enzyme, the ATP synthase, and it adds phosphates to ADP to create ATP. So the protons now are moving back through here, exactly the same motivations as before. It's a chemiosmotic drive to equalize both charge and chemical differences across the molecule. So that's fine. We've created a small amount of ATP, but that is not the singular importance of photosynthesis. The real importance of photosynthesis is that in photosynthesis, two simple molecules, carbon dioxide and water, two inorganic molecules, carbon dioxide and water, are to be bound together to form a complex organic compound. And that is, the initial compound is a sugar, a carbohydrate. What does carbohydrate mean? It means carbon, hydrogen, and oxygen together. Carbon dioxide, CO2, water, H2O. We're going to put those two together and we're going to create something with the formula CH2O, like glucose, C6H12O6. That is a very complex and a very difficult process. And it is one which is anabolic, highly anabolic. It requires, and is highly endergonic, it requires a tremendous input of energy in order to perform it. And all of that energy is going to be derived from here, from these reactions here, from the ATP here, and very importantly from the NADPH. So the first set of reactions occur on the membrane. The second set of reactions are not associated with the membrane, but associated with the interior of the chloroplast if it's a eukaryote, or with the cytoplasm if it's a prokaryote. Remember, photosynthesis occurs in the prokaryotes. It evolved in the prokaryotes, but it, of course, occurs in the eukaryotes as well. But the system used in plants, for example, is very, very similar to systems used in prokaryotes because it evolved in the cyanobacteria, in the prokaryotes. So this whole process takes place during another cycle, a metabolic cycle, which is called the Calvin cycle. And the Calvin cycle is a process which is not dependent on light at all. but which proceeds in the presence of carbon dioxide and of water. And it depends very heavily upon this enzyme here. It has a complex name. It is ribulose bisphosphate carboxylase. And it is abbreviated always to Rubisco. Rubisco is probably the most important enzyme in all of biology. And the reason for that is that we continually lose energy. We lose all of the living world, the entire biosphere. We continually lose energy in the form of heat because all of our metabolic reactions are inefficient. And as a result, we need a continual input of energy. And the input of energy comes in the form of energy from light, which is converted to chemical bonds, the energy in chemical bonds, which is lodged there as potential energy. But in order to create those bonds, we have to create complex organic molecules from these simple building blocks, carbon dioxide and water. So this is going to require a tremendous input of energy, but that input of energy means that energy is going to be stored. And the second thing is it's a very expensive process, very, very metabolically expensive, but so what? The sunlight is free. That is what all of these processes are depending on. They're depending on sunlight, which as long as the sun is shining, these processes can proceed. Okay. The first set of processes we saw, they are light dependent. Those are those membrane-bound processes of photosynthesis which create ATP and create NADPH. Here's where we're going to use them, those products. This Calvin cycle is named obviously for the person who first elucidated it. We're going to see that we're going to take carbon dioxide molecules. We're actually going to take a number of them. Initially, just for simplicity's sake, we're going to say we're going to take 3 carbon dioxide molecules, okay? And we're going to end up creating a compound which we're going to shove around here twice. 2 times 3 CO2s, how many C's? 6. How many oxygens? 6. Okay, we're going to end up with a glucose molecule being spat out every time we do this, every time we revolve this twice. We're going to spit out a glucose molecule. This is the way it's done. This enzyme here, Rubisco, will bind carbon dioxide to this substance called ribulose bisphosphate. That creates a very unstable molecule, 6-carbon molecule, which is going to split. It's going to split up into these 3. You do not need to remember the details of this cycle. What I want you to observe is that we're going to effectively spin this around and around and around, and we are going to continually spit out 6 carbon molecules of glucose there. Each time we spin the cycle, we're going to put in, for each molecule of glucose, we've got to put in 6 molecules of ATP. We've got to put in 6 molecules of NADPH. And here on this side here, we're going to put in another 3 ATPs. That's for a different reason, okay? On this side here, just remember, we're pulling in carbon dioxide. We're going to pull in 6 carbon dioxides. We're going to utilize 6 ATPs. We're going to utilize 6 NADPHs, and we're going to produce a molecule of glucose from that. Very, very, very expensive. We're using a huge amount of ATP and a huge amount of NADPH for each molecule of glucose that is produced by the Calvin cycle. But it doesn't matter. It doesn't matter because we're producing this NADPH and the ATP using free energy. It's energy from sunlight. It is not costing this organism anything to do so. So it is all profit, if you want to think of it that way. And we end up producing these molecules of glucose. What happens to those molecules of glucose? The fate is many and varied. But in producing the molecule of glucose, what we are left with is this 3-carbon compound, which is pushed back into this part of the cycle here and is phosphorylated again with some ATP to regenerate this 5-carbon compound, which is ribulose bisphosphate. Ribulose bisphosphate, 1, 2, 3, 4, 5, plus a carbon from carbon dioxide is 6. That is our first 6-carbon compound. Okay. So that's why this is a cycle. It's a cycle because what we're doing is we're regenerating ribulose bisphosphate there each time we run the cycle. The cycle has to run 6 times in total in order to spit out 1 molecule of glucose. In doing so, it uses up all of this ATP, all of this NADPH. But it's fine because it's free. And the entire biosphere depends upon plants and upon photosynthetic bacteria to do this, to continually convert carbon dioxide and water into this simple organic compound. But the simple organic compound comprises carbons with hydrogens bound to it. In other words, glucose, what is glucose but a highly reduced carbon compound? It has lots of hydrogen atoms associated with it, with all of that energy associated with the hydrogens. And glucose itself, though the energy, the potential energy locked into those bonds is in fact derived from light during the light processes of photosynthesis. Okay, so I will tell you that there is this interactive exercise which I want you to do. On Wednesday, our class will be recorded again, and it will be somewhat shorter than it usually is because I want you to do this exercise, please, extremely carefully. Okay, and make sure that you realize that this is what is contained in that exercise is examinable, but it will clarify for you the processes of photosynthesis. All right, so I will see you again on Wednesday.